Hey guys, this video is going to introduce you to gene regulation and expression. Now, not to muddle the waters, I want to make sure that we understand process over examples at this point. So I'm going to talk kind of in some vague scenarios here just to get you to understand all of the moving parts required. And then in class we can look at some specific examples to understand the complexity and how specific scenarios elicit specific responses. The other thing I want to touch on is types of regulation, negative regulation and positive regulation, because each of these will activate a gene for expression in a different way based on how the environment requires certain things to be produced. All right, so up here you see the basic components of gene expression in terms of reading the DNA for the production of mRNA, because that's where, as we know, protein synthesis really begins. So there's some things. The most important part is the gene itself, the thing that we want to transcribe that should ultimately turn into some sort of phenotype or cellular response. We have the promoter, which is something you've heard about before in reference to the TATA box, and this is where the RNA polymerase lands and begins transcription. Now, the part we really haven't touched on is this idea of an operator, and this operator is kind of the regulatory part of an operon or of a whole gene processing unit, which is what we've got here, which allows something to be transcribed or prevents it from being transcribed based on the environmental conditions. Now, if we want to get into the specific in regard to regulation, the fundamental idea is that if something is not regulated, RNA polymerase binds to the promoter and then continues moving down the gene in order to read it or transcribe it. Now, the operator has the unique opportunity to determine whether or not that happens. So the first type of regulation that an operator can express is negative regulation, and this is where there is the presence of a repressor that prevents transcription. So that is if there is something here on this operator. So the operators have binding sites. So let's assume that there is something that is attached here that is preventing it from happening. And an example of this could be like in the lac operon, or the gene unit that produces lactase, which breaks down lactose. So in normal circumstances, we don't need lactase. We do not need the enzyme that breaks down lactose unless lactose is present. So under normal circumstance, we have negative regulation. We have a repressor that is there preventing this gene, the lactase gene, from being expressed. Now, it is possible, because we do need that. We do need lactase at times, that if lactose becomes present, let's say we drink a glass of milk, it can bind to this regulatory protein and change its shape conformationally, much like in enzymatic activity. And so whenever lactose is present, it can bind to this site, cause a conformational change in that regulatory protein, and then cause it to be removed. And once that repressor, serving as a regulatory protein repressor, is removed, transcription is allowed to occur. But under normal circumstances, there is a repressor that is present that prevents transcription from occurring. Now, the second type is positive regulation, and this would be where something is added that allows transcription to occur. So this would be the addition of a factor that activates transcription. And one example of this, without issuing the name here, is where you normally produce or you normally have or consume, is a great example, a specific protein that your body needs. So let's say that you have under normal situations, you may consume the appropriate amount of tryptophan. But let's say that you haven't eaten something that contains that protein in a particular amount of time. your body does have genes to produce that, but normally does not need to do so. So if you are lacking a particular protein, your body can make it. So in this situation, you have this factor or regulatory protein that normally does not fit into this. And this is not activated at all. So this process up here is not happening. That's because you normally have that required protein that is preventing this from attaching there. Because you don't need it. You don't need that gene to be expressed. But let's assume that for one reason or another, you are not getting that particular protein. When you remove that activator there, this will conformationally change into, sorry about the drawing here, something that would be able to bind here. And when it binds, that activates the process moving forward. So the thing that you really need to consider here is the idea that there are different ways in which genes can be regulated for expression. If a gene is normally not needed, it can either be negatively regulated or positively regulated. And there is not a circumstance where you will need to remember a myriad of different examples, but you need to be familiar with the process in play when it's described to you. So if it is being repressed, that means that there is negative regulation or the presence of something that is preventing it from happening or negatively affecting it. Whereas with positive regulation, it is activated by the addition of a regulatory protein, or it is positively regulated because something is being added or positively affecting it. So for the most part, these examples are, in terms of the structure, are more reminiscent of prokaryotic gene expression. Eukaryotic gene expression is a little bit more complicated, but for the most part, from this video, you need to be familiar with the three parts of a gene, of an operon, or a kind of a whole gene expression unit. And then you also want to be familiar with negative regulation and the general idea, and positive regulation and the general idea. And from there, we can talk about the greater complexities of specific examples and of eukaryotic gene regulation, which is more complicated.